We offered to upload a range of materials that we developed for a Year 1 microbiology course. The reason why we offered to put these into the project was because there were a lot of resources that were already almost built, the idea being that we could convert them fairly easily and quickly into a format that was suitable for OER, i.e. taking off the Leeds branding, making sure that the content was up to date, taking out any images that could potentially have copyright attached to them, redoing them, and then basically repackaging them in a way that would be suitable to be downloaded, whether that be as individual units or as cohesive packages. The older resources were then updated so that they were current and would be usable by anybody that was accessing Durham at this time. Well, they're all housed in Durham Open, which you can see, and all I've done is gone to the Durham Open homepage, and in here typed, for instance this one, it's an introduction to how to use a light microscope, so I've typed in light microscope there, and it brings in all these responses. That one there is mine, so what you would do is you would then click on that, and then you would go down here, and then you just press download, and then that would download, in this case it's a zip file, it would download a zip file onto your computer's hard drive, and then once you've unzipped the file, this is what you will get, and this is a batched up resource. Well, it's all of our resources are fairly generic resources. They're information about basic microbiological techniques spread across, a broad range of not only just microbiology but basic medicine, basic dentistry. So they're not say experiment specific or like time specific. So that's why we thought that these ones would be suitable, because they should potentially have a wide audience that could actually use them. And the way that we just developed them was just taking off anything that was specific. Like we put a lot of quizzes up, and one of the quizzes was a microbiology safety quiz. One of the questions being, where would you congregate was there a fire alarm? The answer for our quiz being Garstang level 6, but obviously that doesn't work if it's a generic resource, so we just took that question out, so just pared down everything that was Leeds specific and just left in all the generic material. Most of the resources that we put up are articulate or engage resources, so they're fairly interactive. A lot of ours were presentations that were made from lecture material, but what we've done is updated them. There's audio files on the back, so it's not only just if you've got the words, you've also they're well illustrated with photographs and the audio file that goes on the back of them. So as opposed to it just being a static presentation, you've got a good deal of explanation that goes on behind as well. Right, well, this took a lot of working out how to do it, and with our articulated presentations, what you have is an articulated version, presentation version, which is like the e-player version, but also what we've included is the original PowerPoint files that we used to produce the articulated presentation. So all the PowerPoint files are then obviously editable. All the images you can take out and replace, if you want to use your own images. And with the Engage files, all the original, because each one of these is a separate Engage file. So we put in the original source file for these Engage interactions that you can then, have you got the Articulate software, you could then edit as and how you want it. So all the original source files are there, all the editable source files are there. There's no branding on, and so they could be used by anybody in any university or any walk of life. Could be used by a tutor to form, the basis of one of their lectures, or it could just be housed in somebody's VLE and used as extra learning resources that can be accessed by the students. This one is part 3 of a series of 3. They've been packaged individually so that you can take each element on their own. Get your copyright sorted out first thing. If I was doing a project starting from scratch to produce resources from scratch, you would know not to have images where you don't know where the source file has come from. You would always use images that you either owned the copyright for or were copyright free, or had permissions from the author or whatever. That was one of the biggest things. And also we've learned how to package things up nicely so that they all flow together and things like that. I think it's easier to develop from scratch than it is to convert. And then just make sure that you include, like we've also included a help file in ours on like how to use the resource and how, if you want to amend it, how to do that. The project that I'm working on now is to develop a load of biochemistry resources, and the remit on the grant application for that was that they had to be available to be released as open educational resources at the end of the project. I think they're generally a good idea. I think the hurdle that I've come across when trying to get people to use them is academics are very keen on owning their own work, and they are quite wary of using other people's work because it's not exactly the way that they would like things to be put across. But that's just, if you want to keep reinventing the wheel, then that's up to you. But there are things out there that you can use, and I am using things that I've come across from doing this open educational resource project, and also things that I've found out there. They're being built into the current project that I'm using. It's a change of attitude and a change of culture, really, that will happen over the next few years. So this is an example that we built which shows you how to use a light microscope. It's got several different elements to it. This one here shows you how different organisms looked after they're stained and viewed under a light microscope. So here we have the actual picture, which you can zoom in and zoom out, and then it has a descriptive text that says how they're stained and what the organism is. There's a variety of these that can be seen. And then another element of this is a video demonstration which shows you how to correctly set up and use the light microscope. So again, there's an audio file on the back of this thing here. So it just basically takes you through the whole setup of setting up a microscope and how best to use it. This one is an annotated diagram whereby each of the individual parts of the microscope is highlighted. So there you have a picture of that one is the eyepiece, which again you can zoom in and zoom out with a descriptive text to go with it. And then the final bit of this, there's a quiz. It's just to test your understanding of the parts of the microscope. So drag these little boxes here, and then you can check if you got the answers right. This is one of the parts of a suite of materials that we put up, which are called the Essentials of Medical Microbiology. There's lots of different parts to it, but this particular one is showing you antimicrobial drugs. So this particular slide is on antimicrobial chemotherapy, and this is a very basic slide just with pictures, but there'll be an audio file on the back talking about what these particular drugs do. Going further down the interaction, these are just basic cell wall components, but again in the background there will be speech going on telling you what the importance of these bits are, and that can be accessed in this notes panel here. So if you're hard of hearing, you've still got the information showing on the notes panel there.